everyone. Welcome back. My name is Sarah Kolb, and I'm your host of Learn and Burn. On this show, we have scientists' questions on life science technologies as they attempt to conquer a gauntlet of hot sauces. Today's topic of conversation, Lumit, a new immunoassay technology with fast and quantitative results. Today, I am joined by Hicham Zegzouti. Hicham is a senior research scientist and a group leader who has been at Promega for almost 14 years. He develops biochemical and cell-based assay technologies to integrate diverse enzyme activities with cellular pathways. He leads the group at Promega charged with developing new methods of protein and post-translational modification, detection, and advancements in immunoassay systems. Welcome.
Thank you, Sarah. Nice to be here.
Excited. Ready to taste some hot sauce?
Well, I'll see. As a Moroccan, I am not used to eat like hot sauces, but we usually eat a lot of spices in Morocco. But I am taking the challenge,
Well, good luck. We'll see how we do.
Yes, let's do it.
Each of these sauces have been made from locally sourced produce grown in the garden on Promega's campus in Madison. So let's get started. All right. So our first one is the habanada. So I'm going to take this, douse my whoops in it. All right. Cheers.
Cheers. Okay. This is good.
So how does this one compare to your traditions?
No, actually, that one is still good in terms of not hot. It's even sweet. I'm going to add some more, actually.
Mm-hmm. Yeah.
So up to now, I'm still good, It's a good start.
Yeah. It's a smooth entrance.
Yeah, definitely.
All right. So now that we've had our first couple bites, can you give an audience a brief introduction to Lumit immunoassay technology?
Yes. So Lumit technology is actually a homogeneous immunoassay that we developed in the last 2 years. We are trying to detect any analyte by using a homogeneous setting. So it means we actually, if you are familiar with our nanobit technology, which is a nanolock luciferase complementation system, we actually label two antibodies that recognize two different epitopes on the same analyte. That could be a protein, for example, of interest. And those two antibodies, one is labeled with the large fragment of nanolock, which is large bit. The other antibody is labeled with the small fragment, which is small bit. So when those two antibodies find the analyte in the tube or a well, we use a assay plate. When they find the analyte, both of them are on the same protein, and it means they are bringing the 2 nanobit moieties or subunits into close proximity, and that actually create an assisted complementation that generates luminescence. So what is good about this technique is actually you don't have to, for example, like ELISA or Western, you have to run a gel, transfer, or for ELISA, you have to coat the plate, etc. So it's heterogeneous because you have to do a lot of washings. This Lumit technology is actually homogeneous. It's in solution assay. So it means you add your sample and you add the antibodies, and then you add the furamazine, which is the nanolock substrate, and you detect light in a simple luminometer. So it's in solution based, very easy. It takes from 30 minutes to 2 hours, depending on what application.
Ready to indulge in our second wing?
All right, let's see how it goes.
So this is the Ahi Rico.
Yep. Okay.
How do you think this one's going to go?
Let's see. It smells good.
What are some of the hottest spices that you use?
Oh, so we use the cayenne, a little bit of it. We don't have sauces based on that. But what we use is the sauce, for example, that is not originally from Morocco, but it's from Tunisia, and all North Africa actually adopted it. It's called Harissa, if you heard about it. So harissa is a pepper paste with garlic, so that gives it that good flavor. And
Mm.
that's basically the hottest that I use, or even all Moroccans use, except what I said in the beginning. We use spices like cumin, paprika, cayenne, etc. The only memory I have of hot pepper in the family is moms and aunts. You see them during like lunch or dinner. They have always a plate with just a fresh jalapeno, and they eat whatever they are eating, and they take that one and they take a piece of it in their mouth. But usually we're impressed because we couldn't do that, so it's hard for us.
Now that we've had a little bit of hot sauce, can you tell us more about the workflow for Lumit and what does that look like? And, does the assay itself take different forms?
Oh, yeah, great question, Sarah. So, yes, so when we developed the Lumit, we had in mind that it's going to be used in many applications. So, as I said, it's an assay that is homogeneous in solution. You don't have to wash or do any step. Just add solutions to your sample. So the way we set it up is first labeling primary antibodies, for example, to detect directly an analyte in solution. So that's common knowledge in immunoassays. The second one is indirect, where actually we take advantage of the availability of different primary antibodies. So we labeled secondary antibodies with the nanobit fragments. So now if you have an analyte where two antibodies are available to bind to it, one from mouse, one from rabbit, for example, we use Lumit labeled secondary antibodies that are anti-mouse and anti-rabbit, and it creates the same complex and generates light. The third application that another group at Promega is using is competitive assay. So basically it entitles you to label a tracer, which is, for example, the assay that we have, that they have developed is for FcRn binding. So FcRn is a protein that binds antibodies, so you can screen for that binding application. So you have an antibody that is an FcRn that is already labeled with one part of the lumits, and then you have the second part is the antibody. And if there is a competition between the antibody and FcRn, you can see a decrease in light. So that's the third one. So, and again, so it's an homogeneous assay, and the format is a plate format where you have just volume ratios. For example, for indirect assay, you just add your analyte that will be in the cell. I can talk about that also later. Cell lysate. You add the antibodies in same volume, and then you add the ferromazine, and you go to the luminometer and you read the plate. So it's a very easy and fast workflow.
Mm-hmm. Sounds like Lumit knows how to bring in the heat into the lab by making things
Definitely, we
more
could see that.
fast and fun and get
Yeah,
research going quicker.
exactly, Sarah. So that goes with the heat that we're going to be experiencing probably in the third one.
Yeah, which is funny because the third one is called liquid lava.
Oh, it goes
So,
with homogeneous. Yeah. Solution based. Okay, let's do that.
so I'm thinking that I'm going to start feeling the kick. I'm
Okay.
starting to feel it a little bit like on the edges of my
So,
mouth.
oh, I haven't actually used the milk yet, so probably after this one. So I put some, I'm not using wings today. I'm using just a chicken strip. So,
Right.
okay. Like, oh, yes. Oh, yes. Probably we're going to start seeing fluids coming off the eyes.
I put a little too much on.
Me too.
Woo!
Mm.
I just feel like
Wow.
the jump from the ahi riho.
What happened is a big jump.
I mean, we went from 17,000 Scoville units to 60.
Wow.
And now that we We're definitely feeling the heat come.
Mm-hmm.
Let's talk about one type of Lumit immunoassay that has been developed specifically to interrogate cell signaling and pathway nodes. So sorry.
I hear you. I feel you, actually. Oh, this is definitely starting to kick.
Yeah.
Well, actually, it's hot, and I'm going to talk about the hot area. So the immunoassay, the Lumit immunoassay cellular systems, as its name mentions, we actually look at phosphorylation of different signaling pathway nodes in cell lysate. So the way we do it is actually we take cells that we, for example, activate the pathway of interest, or if you have a cancer cell where you have that pathway already, active, and we select a protein or that we call a node in that pathway that can give us like a reporter. If it's phosphorylated, it can be a reporter for the pathway activation, or for instance, for drug discovery, for inactivation or inhibition. So an example of that, if you take the MAPK pathway, it starts by activating the receptor like EGFR, for example. Then you have RAS protein that gets activated, it's GTP bound, and that triggers different effector signaling downstream through different kinases. So it's a MAPK pathway, it's a cascade of different kinase phosphorylation until it reaches ERK, which gets phosphorylated by MEK. And then ERK goes to the nucleus to phosphorylate transcription factor to, of course, transcribe genes that are involved in multiple biological processes. So that pathway is, can be mutated in different cancers. So in order to look at that pathway activation, for example, if you have a RAS mutant that is all the time active and you want to inhibit it, you can use phospho-ERK as a reporter for that pathway inactivation. So you just treat the cells with the inhibitor and look at treated versus untreated cell. And if you see phospho-ERK up with the inhibitor, it should go down, so you will not see it. So we use this Lumit immunoassay cellular system, but because we wanted this assay to be universal, we created that secondary antibody concept where you have two antibodies that are not labeled against ERK and phospho-ERK, of course. And then we use secondary antibodies. And this way you can apply this technology to many, many other targets.
It's funny, while you were sharing that, I just kept thinking about, how the technology can be used in so many different applications, but then how every time I eat hot sauce, no matter what it is, like my nose just keeps
Yeah. Well,
so I'm like, wow, that's universal, kind of like this technology, kind of cool that it can be used in so many different ways, like clearing out my sinuses.
Definitely. I'm sure there is a pathway there that gets activated or probably inhibited now by the capsaicin in the pepper.
So Hisham, we're just over the hurdle. We only have 2 more sauces to go. How you feeling?
Well, actually, it's not what I expect. I expected to be already crying. I have my Kleenex here with me. So usually my friends know me because every time we go, I have some Indian friends, so they have spicy, hot food. And every time they know me, I start just the first bite and I take my Kleenex and I start, Right now, I'm really feeling good because I thought I'm going to be doing that already, but I am good. So.
This sauce is called the Hulk.
Whoa. Mm. But you don't feel it in the mouth. You feel it here in the throat.
Yep.
Oh. But again, I like it actually. I'm gonna add some more.
Uh-huh.
I like it. Even if it goes into the throat, I have my milk, so I will use it later. But now.
Yeah.
But
Oh
still,
my gosh.
I'm gonna go with the milk.
Oh
yeah.
Whew. Now that we've enjoyed the 4th sauce, I'm clearly starting to feel it in the lips, in the mouth, in the back of the throat. What are some other applications of the Lumit technology? I know, Hicham, you mentioned earlier that there was some more. Could you share with us what those are?
Yeah. If I can get out of this trance state.
The water helps at first, and then afterwards it shoots the tongue on fire.
And when you take air into your mouth, it makes it even worse. So, okay, I'll try to compose myself and tell you about the applications. That's what you asked. Yeah. So the Lumit technology now, it's being applied to many detection systems. So we have a group, like I said, my group developed the Lumit immunoassay cellular systems. We are validating signaling targets. We will be also looking at different post-translational modification, not just phosphorylation, using the same system like secondary antibodies that go with non-labeled primaries. So the secondaries will be labeled with Lumit. Then another group in our assay design, bigger group, they are developing Lumit assays for cytokines. So they have a program where they are developing, they have an assay for IL-1 beta cytokine, for IL-6, for many other interleukins that are important, of course, in terms of these proteins actually are secreted, so it's a different application. You are not looking at them inside the cell, but outside the cell in, for example, media or serum, etc. And then the third kind of assays, we have another group developing insulin and glucagon and Lumit immunoassay also. And then I described the FcRn. So the FcRn is one assay that was developed already for biologics. And then they are going also for different Fc receptors like the gamma receptors, etc. So those are the ones that we are in process of either already launched or we are in process of developing.
Okay, Hicham, last stop.
Yep. Oh, I'm expecting the worst here.
We've made it this far together. I expect by the name of the sauce, Fatality, trying to go chug some sour cream or whatever.
Okay, I'm going.
Let's do this together. This one, it lingers, and then it's pow.
Yeah, I'm waiting for it to linger now.
Yep.
Yep.
There's the pow.
I'm almost doing it slowly, like meditating while I'm doing it.
Getting in the right mindset to eat a very intimidating hot sauce.
Well, remember I promised you some crying. Not yet, but I'm just gonna use it. Excuse me.
Okay, let's see if I can even ask the last question.
All right. Fire up.
Whew. Hicham, please give us a shout out to your latest news around Lumit.
Yeah. Okay. So we are actually doing some hot things right now. It's so we are looking at remember this year it was not a hot year, it was bad year during the pandemic, and we wanted to contribute also to the science going around COVID-19. So during last year we developed an assay based on Lumit where we looked at the interaction between the SARS-CoV-2 spike protein and its receptor on the cell, human cell. So those two proteins are called RBD, which is receptor binding domain. That's the viral part. And then the ACE2 is an angiotensin converting enzyme number 2, which is on the cell. So we took those two proteins and we wanted to create a PPI assay without touching the proteins themselves, not to interfere with the binding, and use that for screening molecules that can disrupt that interaction. So the way we did it, we took an FC tagged RBD, the FC from a rabbit, and an FC tagged ACE2, and that FC is from mouse. And we took advantage of our Lumit secondary antibodies. So we use an anti-rabbit small bit and anti-mouse large bit. And now you have a complex that is between, instead of having two primary antibodies and the Lumit secondaries, now you have just the proteins with their FC domain or the FC tag. So the FC is basically like one part of an antibody that can be recognized by a secondary antibody. So the assay works really well. So what we did with it is during this last year, we apply it to screening anti-spike protein antibodies. So we had a mini set of antibodies that were developed against the spike protein. So we found different profiles of inhibition, like antibodies that disrupt the PPI. Then we took the assay and we used it to screen patient-derived serums and plasma samples from infected patients with COVID-19 to look at neutralizing antibodies. So how the serum, constituents like the IgGs that were developed after the infection, how did they neutralize the PPI in our RBD ACE2 assay? So the third thing that we did is we took like a small set of peptides that were published before to recognize RBD, and we tested their potency against this interaction. And then the cool thing that we did is we took that RBD and we created or generated different mutants of it corresponding to the variants that you hear about, the virus variants. And we plug them into the assay to screen for antibody inhibition escape. As you know, people are interested to know if you develop antibodies, are they going to be potent against only the wild type virus, or also they can inhibit the variant mutants or the mutant variants. So we changed that RBD to the mutant variant and we tested again those antibodies, and indeed we found that some antibodies actually escape now the inhibition. They don't inhibit the mutant. So that's something that you when you find it, you need to find an antibody that can actually inhibit all the variants plus the wild type, of course. The last application that we did with that is this is a collaboration that we did with the University of Rio de Janeiro in Brazil. They have a large cohort of patient serums and also vaccinated patients. So we took those vaccinated serums and we tested them, and they range from multiple days before first dose, between the first dose of vaccine and the second dose, and then after the second dose. And you can see a beautiful spread of neutralizing antibodies that increase after the first dose and dramatically increase after the second dose. So this assay actually allowed us to have a proof of concept for looking at PPI using Lumit technology. And you can apply it for other virus-human interactions in the future. Mm-hmm. So other people can take the same concept and use two proteins that interact and see if they can disrupt that interaction with antibodies or compounds.
It's interesting because you hear the names of technology, right? You hear Lumit, and then it's really fascinating to learn the impact that Lumit has in what everyday non-scientists and non-researchers can relate to, right? Like you said, the pandemic has impacted us all, Definitely. Cancer impacts us all, we always, we don't always realize the name Lumit might have all of these repercussions for advancing discovery. So thank you for sharing. That is awesome. And definitely a very hot topic.
Yes.
No pun intended.
Yeah, no pun intended. I'm happy that I didn't die yet. I'm still good. But I like those sauces actually. So I at least now I can put a number in my head. So I can go up to 100. You said 25,000?
Oh, thank you so much, Hicham, for joining us today and sharing about Lumit technology and enjoying these extremely fun sauces, I'll call.
Thank
And
you,
always remember to stay sizzling.
Oh yeah, I will. Thanks, Sarah. So yeah, I'm gonna go have a nap. I'll finish probably my lunch and go have a nap.
Finish the chicken tenders.
Exactly.